So today we will talk about complementation, and we have talked about this topic in great detail when we were talking about epistasis, and we also talked about how the purple color plant and how when the purple color plant was mated with a white color plant and what were the uh, outcomes. So we looked into that there are the formation of a purple color at last is a combination. It's a multi-step process, or it was a two-step process where precursor was there and there was an intermediate, and then intermediate was converting into purple color color product. So now we will talk about how this complementation work and why complementation is important, and how complementation helps till date to get to know that how many genes are involved in particular pathway. We know that a product is the product that we see at last in biological system can be a multi-step is a result of a multi-step process. You can relate this very well with the glucose, where a glycolysis pathway follows nearly 10 steps and pyruvate is formed at last. So if anything went wrong in between, then the product, which is pyruvate, is going to be hindered. The formation of pyruvate is going to be hindered. So how many steps are there, which genes are involved in it, are can be can be deduced through complementation pathway. And lastly, we will talk about that how complementation process is different from recombination. We know that the complementation can also give a new phenotype, and whereas recombination also gives us new phenotypes. So how these two processes are different: in one there is no physical breakage of chromosome occurs, which is complementation. In complementation, you will look that there is no physical breakage of chromosomes are there, but when you will see the recombination, you know that the crossing over phenomena is there, and there is breakage of chromosome part occurs, and there is some exchange of chromosomal segments occur. So we'll look into that also, but first we'll start with complementation. Now, what is complementation? A journal definition of complementation is the ability of two mutants in combination to restore a normal phenotype. So let's suppose a normal phenotype is presence of purple color. Okay, so the normal phenotype is presence of purple color. So gene A helps to convert a precursor into intermediate. So there is one gene which codes, so gene A, which codes for enzyme A, that will help to convert a precursor molecule into an intermediate molecule. Now we have intermediate molecule. There is other gene B, which is going to code enzyme B, and that will convert intermediate phenotype or the intermediate product to final product, which is purple color. So now the purple color is a product of is a product of two gene or the two enzyme, where first a precursor converted to intermediate and intermediate converted to purple color. The main thing that you have to bring in your notice is that if any of the path, if any of gene like gene A or gene B gets mutate, if gene A or gene B gets mutate, then you are not going to see purple color. So in first condition, so we'll take first condition where gene A is normal, gene B is mutated. Because gene A is normal, it will code for enzyme A, and enzyme A will help in conversion of precursor molecule to intermediate. Okay, but gene B is mutated. Gene B is mutated. So enzyme B will be mutated, and it will not be able to convert an intermediate product into a final product, which is purple color. So the take home message from this line is the take home message in this line is, if any of the gene is going to be mutate either A or B, then it's going to hinder your final product formation. Okay, But in other case, when you will see what I'm trying to, so in other words, what I want to make you understand that if, so this was a normal example that we looked into it, and let's suppose there is one organism. So I'm taking a little different example now, so to relate this directly to what we are going to look in upcoming slide. So let's suppose there is organism A, oh sorry, organism 1 and organism 2. Organism 1 has two genes for formation of purple color. Similarly, organism 2, so like this, let's start like this. So this is organism 1, this is organism 2. It has two genes A and B. Okay, so organism 1 is a haploid organism. We know there are various lower organisms which remain in the haploid state, such as fungus or fungi. So organism 1, which is in a haploid state, so it has two genes A and B, to which gives you the purple color to fungi. Let's suppose purple color to fungi. This is an imaginary example. Similarly, organism 2 or fungi 2 also has A and B gene, which gives you purple color, because organisms are same, so the genes will be same. What I'm trying to say is, let's suppose in the organism 1, which was haploid, because we know that the fungi can remain, they can be in two state, haploids, and then haploid mate and they form diploid. So before sporulation, the fungi remain in diploid state. So what I'm trying to make you understand that the fungi 1, which has two gene A and B, and they together work to give you purple color to the fungi. Similarly, fungi 2, which has A gene and B gene, they are working together and they are giving you the purple color. Let's suppose the gene, so the organism 1 is, let's suppose, is mutant for A gene. So it was mutant, so I'm removing capital A, and for B they are normal. Okay, organism 2 is mutant for B, whereas A is normal, so B is mutant. Okay, so 1 is haploid, 2 is haploid, they will make gametes, and then they will become haploid gamete, and haploid gamete they will fuse to give a diploid fungi. Now this diploid fungi, so the fungi 1, which was having which was harboring a mutant A gene and one normal B, and we know that when capital A and capital B will are there, then only the color of fungi will be purple. Similarly, for fungi 2, when the capital A and capital B are there, then they will be purple. But in both the cases, A first has a mutant gene for A, and second fungi has a mutant gene for B. So this means that they both are not purple. They both are not purple. Let's suppose they are white. They are white. They are in white color. I'm saying again and again that it's an imaginary example, and this example has been taken just to correlate the things. But when they will fuse, when their gametes will fuse, so from first haploid fungi, it will be having small a, capital B, and it's diploid. So from second fungi, it will be capital A, small b. So what will be the color of this fungi? White or purple? It will be purple because here we have a normal gene, capital A, and from one we can get capital B. They can complement each other. They can, so A will help to convert a precursor to intermediate, and B will help to convert intermediate into a purple color product. So what I am trying to say now again, read the first line. A general definition of complementation is the ability of two mutants. So here two mutants were haploid fungus 1 and haploid fungus 2. They were the two mutants in combination. So when they will be in combination, when they will be in diploid state 1 and 2, to restore a normal phenotype. Normal phenotype was purple color. So here we got purple color. So again, read the definition. A general definition of complementation is the ability of two mutants in combination to restore a normal phenotype. Complementation distinguishes between mutation in same gene or in different gene. So we will look into this, that how this process helps us to define that the mutation is in the same gene or it's in the different gene. So how complementation is helping us? So it is giving an idea that the mutant that we are getting, so let's suppose, because two genes are working together to give a purple color, we get mutant. There was a mutant there. So let's take an example. So fungi number 1 was white because of some mutation in A or B gene. Similarly, fungi number 2. So it's a different example. I repeat, I'm taking now a different example. So fungi number 1 was white because of some mutation. Fungi number 2 is also white in mutation, either in A or B. We don't know. So because this was our condition 1, this is our condition 2. So this is condition 2. So in condition 1 we saw that when the two mutants when they were combined, they gave a normal phenotype that was purple, which was purple. Now here in case number 2, when one number when one of the haploid fungi, which was white, and other fungi number 2, which was also white, when they were when they mated and they made a diploid state, when they made a diploid state, the the system or the fungi remain white. The fungi remain white. Here the complementation did not occur. Here the complementation did not occur. It might be possible that both 1 and 2, they 1 was also having mutant allele small a, and second was also having a mutant allele small a. So because both were having small a, so the precursor was not turned to intermediate, although both were having a dominant or a wild type allele capital a wild type gene capital B. So both were having capital B, but what will be the use of that capital B? What will be the use of that capital B? Because capital B will act on intermediate. That intermediate is absent here because both the mutant 1 and 2, they are having a mutation in the same gene that was small and they are having the same gene which was small a. So this means that if the mutation is in the same gene, then the complementation is not going to occur. So what I will suggest you, that in this second case, I took mutant capital or small a, small a. You can reverse, you can take mutant small b, small b. So then see what will happen. So in this condition, the precursor, because we have just discussed that both 1 and 2 fungi, first and 2 fungi, they both have capital A, but they both have capital A, but they both have mutant B, which was small b. So here precursor will turn to intermediate because of capital A, but there is no B. So intermediate will not turn into the final product, which was purple. So this means here both the mutants 1 and 2, they were having mutation in the same gene, which was small in gene B. So I will request you to read the line 1 again. A journal definition of complementation is the ability of two mutants, so mutant A and mutant B, in combination to restore a normal phenotype, because mutant A is giving normal B, mutant B is giving normal A. So mutant A, which was giving normal B, and mutant B, which was giving normal A, when they will meet each other, their products can they can do handshake with each other, and then they can lead to formation of purple color. So come to line second. Complementation distinguishes between the mutation in the same gene or in different gene. I guess now it will be very much clear to you. The ability of third line, the ability of complementation analysis to determine whether mutations are in the same gene or different gene, is the basis for genetic dissection. Dissection means when you are uncovering things, when you are discovering things, when you want to know that how many genes are involved in a particular pathway. So in this process, one finds the gene whose products are required in a pathway. So you can correlate now that the formation of purple color was dependent on two genes. Okay. So likewise, you can also, so people have through this complementation mechanism, people have found that there are like 4 gene or 5 gene or 6 genes are required in a particular pathway. So people have worked on it. So in the example from peas, so we know that for the formation of yellow color, for the formation of color and the formation of this round shape of the pea plant, pea seed, there are two different pathways. So in the example from peas, the metabolic pathway to yellow pigment. So metabolic pathway, you can, so I'm taking, so to make you understand what are metabolic pathway, you can correlate it with the glycolysis pathway. Glycolysis is a metabolic pathway. So the metabolic pathway to yellow pigment is distinctly different from the pathway to round peas. So which is the starch biosynthesis pathway. So complementation analysis is useful in dissecting the steps in pathway starting with many mutants that generate the same phenotype. This is a more conventional example of complementation. So this a very important line comes here. A very important line comes here. The many mutants that generate the same phenotype. So mutant 1, so in this case, in our second case, where the mutant 1 and mutant 2 were giving white phenotype. Similarly, this was the second case. In third case also, the mutant 1 and mutant 2 was giving white phenotype. So mutant 1 and mutant 2 was giving white phenotype. So this was our second case, where mutant 1 was also white, mutant 2 was also was giving white, and in third case here also the mutant 1 was white, mutant 2 was white. But we know that in both the cases, in this case the defective gene was small. So the defective gene that they were having was small a, and in this case they were having small b. But in our first case, where the mutant 1 was having a normal capital, so the mutant 1 was having small a but normal b, mutant 2 was having capital A but a mutant b. When they mate each other, they in deprived state, they gave purple color. So read it again. Complementation analysis is useful in dissecting the steps in a pathway starting with many mutants that generate the same phenotype. Here in second case, both mutant and mutant 2 are giving you the same phenotype, that is white. Okay, in this case the mutant 1 and mutant 2, they both were giving you the same phenotype, which was white. So you can correlate this when I will take one example. So then what I'm trying to say is that, let's suppose you generated 10 mutants. You generated 10 mutants: mutant 1, mutant 2, mutant 3, mutant 4, mutant 5, 6, 7, 8, 9, 10. and all these 10 mutants are giving white color. That is the same phenotype. All are looking white. 10 mutants, all are white. Okay, because we know that the final product formation, in case, let's suppose in case of glycolysis, the final product formation is a 10-step process. So if any of the enzyme gets mutate, then the final product is that there is no pyruvate. Let's suppose that there is no pyruvate. If enzyme 1 get mutate, then no pyruvate. Enzyme 2 get mutate, no pyruvate. Enzyme 3 get mutate, no pyruvate. Enzyme 4 get mutate, no pyruvate. Similarly, enzyme 9 get mutate, no pyruvate. So what I'm trying to say is that we generate a hell lot of plethora of mutants that are giving the same phenotype, and this is a more conventional example of complementation, and we will look into it. I guess if you leave the last line, don't get confused. What you have understood till now is correct. When the two things, so one is, so let's suppose a two country army are there and they want to defeat a very powerful army, which is, so first country, let's suppose one country, the weak countries, so one country has a very good air force, second country has very good naval uh, naval strength, but the enemy country has both the things, good air force and good naval. So these two weak countries can only defeat the enemy country when they will come together, because one can provide a good air force, one can provide the good naval, then only they can defeat the enemy country. Similarly, the mutant one, so mutant one of case one, it was having a normal B, and similarly the mutant 2 was having normal A, and then they only gave a purple color. So I guess this complementation pathway thing is clear. So now I guess I will be more clear to you. What we just discussed in previous slide, I am going to make it more clear by rereading these lines. So many fungi can propagate as haploids, but can also mate to form diploid prior to sporulation. So this was the example that I was talking in previous slide, and I request those students who are not able to understand the haploid state and diploid state, they should go and they should refer their class 11 book chapter 2, where the fungi life cycle is well described, where they have shown that how haploid fungi, they form a diploid, and they get into their asexual cycle. So there are two cycle of their life, asexual and sexual. So I guess you know this, and you can very well correlate all these things. So thus one can screen for mutants in haploids and obtain recessive mutant, and then test their behavior in combination with other mutants in the diploid state. So that was exactly we just talked in our previous slide, where in case one, I remember the case one, so mutant one, which was haploid, the gene, so they were white. Mutant 2 was also white, but when they went into a diploid state, they were purple. So the color was purple. So what I want to tell you is that in case the mutant 1 was, the defective gene was A, in mutant 2 the defective gene was B, because of which they both were white, but when they came in a diploid state, then they became purple. So they were complementing each other. So mutants were complementing each other, mutant 1 and mutant 2, when they came together, because they were having one half and other was having, so mutant 2 was having other half. So when they gave a very, so they complemented each other. Let's say that a haploid strain of fungus was mutagenized and screened for arginine auxotropes. So there is very important term comes here is auxotroph. Very important term, auxotrophs. So auxotrophs are those uh, organisms where they are not capable of synthesizing a particular chemical, which is very important for survival. So in this case, I am taking a mutant that require arginine to grow. So the fungi, the haploid strain of this fungi is very much capable of synthesizing everything which is needed for their survival. But a very important chemical, which is arginine, we know that the arginine is one of the amino acid, and it's a building block, so it will be very much needed for the normal development of the fungi. So they are just not only able to synthesize arginine, so they are dependent on arginine from outside, from media. We are giving arginine to these fungi who are mutant for arginine, who are arginine negative, so that we give arginine from outside, and their needs are done, and they can very well grow. You can just correlate this with a thyroid case of a thyroid, where we know that when after certain age group, or because of some medical reasons, a person is not able to synthesize a thyroid hormone, because of which like uh, we have seen that when this thyroid hormone is not synthesized by the body, the people or the person or the individual who is suffering from this deficiency of thyroid hormone in his or her body, they suffer from and they have various kind of pathological manifestation. But when we start giving them thyroid from outside, they start taking thyroid pills, and their needs are very much taken care. And so the need of thyroid can be fulfilled, and those symptoms which were there prior of giving thyroid pills, they will they will go. So, and you can also correlate it with the diabetes, where a person is not able to synthesize insulin, and we give some insulin shots, and then the person is very much normal. So a person is oxytrophic for insulin in this case, or the person was oxytrophic in for uh, for thyroid pills in previous case. So I guess now you are able to correlate that here the function, the fungi is very much well capable of synthesizing everything which is needed for their survival, but they are short, or they are mutant for arginine biosynthesis. So we have to give arginine from outside. So those strains will be called so in this case they will be called as arginine oxotrophs. But those mutants are so but then the fungi, so the fungi which have everything, including the arginine biosynthetic biosynthesis formation gene, which is normal in them, they will be called as prototroph. So if so the function, so the motto here is that when one mutant which is arginine oxotroph, and other mutant which is also arginine oxotroph, remember we just talked that we are going to create a 10 mutant for the same phenotype. So here the mutant 1 is also arginine oxotroph, the mutant 2 is also arginine oxotroph. They are giving the same phenotype. I'm repeating the same phenotype. The same phenotype is the arginine oxotroph. So we are creating the same arginine oxotroph. Now we want to see that the mutation in mutant 1 is was in and the mutation in mutant 2 arginine oxotroph. So those mutations were in the same gene, because of the same gene mutation they were arginine oxotroph, or because of some different gene they were arginine oxotroph. Read the third line. Six of the mutants were mated to form the possible diploid combination, and they tested for the ability of the diploids to grow in absence of arginine, that is prototrophy. So in previous case, when we were talking about in case 1, when the mutant 1 and the mutant 2, they were white, but when they were mated, when they were in diploid state, they gave purple color. So they were complementing each other in diploid state. Similarly, what we are going to do here is that we are going to create the 6 mutants, and 6 mutants, they will be short of arginine biosynthetic biosynthesis pathway in their body. So they will be called as arginine oxotroph. The phenotype is same. They will not grow in the absence of arginine. They will require arginine. But if I combine any of the mutant, let's suppose mutant 1 and mutant 3, when they because the mutant 1 is also haploid, mutant 3 is also haploid, I just said that we are creating 6 mutants, and all 6 mutants are giving same phenotype, that they are arginine oxotroph. I mean that the mutants that require arginine to grow. What I'm saying is that I took mutant 1 which was haploid, mutant 3 which was haploid, and they were arginine oxotroph, but when they mated, when they were in diploid state, they were they were very much self-sufficient to synthesize their own arginine. They were not no more dependent on arginine from outside. So they were in prototrophy state. So we are looking into those into this field. So because the mutation in mutant 1 and the mutation in mutant 3, they are not in the same gene, because if the mutant 1 and the mutant 3, if they have the mutation in the same gene for the arginine biosynthesis pathway, so what I want to make it clear again and again, that the arginine biosynthesis pathway is not a single gene, is not dependent on single gene. There can be 4 genes which are involved in arginine biosynthesis. We are deciphering. I'm assuming that, let's suppose there are 4 genes which are involved in arginine biosynthesis. We want to decipher that which all are those genes, and how many genes, because I took that, let's suppose there are only 4, there can be 6, there can be 3. So we want to know that how many genes are involved in arginine biosynthesis. So mutant 1 and mutant 3, when they mated, they gave a diploid state which was prototrophy. They were not requiring arginine from outside, and this means that the mutation was on some different genes. So the gene, because so the mutation were on the different gene, they were not on the same gene. If the mutation in the mutant 1 and the mutant 3 was on same gene, then they will remain in a auxotrophic state. So these things will be more clear when I will show a picture in next slide. The results are tabulated in next slide with A, that is plus, designating growth in the absence of arginine. That means that the plus are in prototrophy state. The plus are in prototrophy state, and the minus are remaining in auxotrophic state. Okay, designating no growth.